Thank you for that introduction, and thank you for allowing me to present and share my work with you today and showing how listening to your nerves can actually promote immunity in your skin to fight parasitic worms that try to infect your body. But before I get into this concept, I wanted to share with you an idea that has actually intrigued me for my whole scientific career, which is how peripheral tissues can actually maintain homeostasis by controlling the extent of inflammatory responses. When tissues encounter pathogens, for example, they need to quickly enable efficient inflammatory responses that are able to prevent the replication of these pathogens or dissemination throughout the body. However, these same inflammatory responses, they need to be controlled. Otherwise, excessive inflammation can actually lead to a lot of tissue remodeling and immunopathology. So understanding this tissue homeostasis requires this balance. Maintaining this tissue homeostasis requires this balance between low and high inflammatory responses that enable not only maintaining tissue homeostasis, but also preserving organ function. And I've been quite interested in how these mechanisms happen. And I've been using parasitic helminths to understand these responses. And in the first part of the talk, I'm going to show you how understanding the biology of these pathogens can actually shed light into how to control excessive inflammation in peripheral tissues. So parasitic helminths are actually useful for understanding this, not only because they're actually pretty cool, as I'll just show you here in these pictures on the left, showing the diversity in the morphology of these parasites, but also in the fact that this diversity is also reflected in the different ways that these pathogens can actually infect mammals. Humans can actually be infected by helminths by ingesting infectious eggs or by the direct penetration of the larvae into the skin or aided by mosquito bites. And it's in these first steps of infection where parasites have evolved different ways to actually suppress, to silence those inflammatory responses. Therefore, understanding this biology can actually shed light on how this immunity is quickly initiated. But after suppressing these mechanisms, helminths quickly disseminate through using the blood circulation into different peripheral tissues and establish these chronic infections in either lungs or the intestine that many times can actually go unnoticed by the patients. So I was actually quite intrigued to understand how these chronic infections can be controlled in the context of a chronic infection with helminths. And to do so, during my PhD, I used an animal model for human hookworm infections here shown on the left. And again, in the lab, we actually were able to replicate the life cycle of this parasite by inoculating this larvae into the skin of mice. And after they reach the blood circulation, they quickly arrive into the lungs and they induce these vast amounts of tissue damage, as shown by the picture on the right, showing this high hemorrhage in the lung. And this actually results in damage of the epithelium here shown in the center, and this inducing the release by these damaged epithelial cells of inflammatory mediators known as cytokine alarmins, such as IL-33. The signals are quickly sensed by tissue-resident innate lymphoid cells or ILC2s, and in response to these signals, they secrete other type of inflammatory mediators known as type 2 cytokines. These molecules now are actually quite effective to act again back in the epithelium and promote mechanisms to fight this infection, such as the production of mucus that is able to remove the parasites from this tissue. But at the same time, these responses actually enable the healing of the tissue, and this is again exemplified here by this picture on the right, showing how quickly the tissue can heal by the action of these type 2 cytokines. And the field has done incredible work on understanding how these responses are initiated. But when I started my PhD in Rutgers, I wanted to understand how these responses are also contained. Otherwise, as you can imagine, excessive production of these cytokines can actually result in the continuous remodeling of the tissue, preventing organ function. And during my PhD, we found that a neuropeptide known as normetin B that is potentially being released by lung innervating neurons can directly act on these ILC2s and function as a brake and telling these cells to stop producing type 2 cytokines before they actually start secreting many of these factors and compromising the integrity of the tissue. This communication between ILC2s and normetin B is actually enabled by basophils that only enter the tissue once the parasite has been cleared. And in the lab, we found that, for example, if we break this circuit, for example, by depleting basophils, this actually results in the excessive production of the cytokines that results in the excessive production of mucus, as shown in this picture on the left, on the right, where the airways are actually blocked by all the tissue, by all the mucus, as well as the cellularity increase in the absence of basophils. However, turning on this mechanism is actually detrimental, as shown here on the left, in which we started activating this circuit way too early before basophils enter, and this actually compromised now the healing of the tissue. So I hope you can appreciate by these examples that the timing of controlling this inflammation is essential to maintain the integrity of the lungs, and this is actually enabled by peripheral neurons. So then I moved into my postdoc, and I wanted to ask if the same regulatory role was of neurons in tissues actually happen in the first initial stages of the infection by controlling these first initial inflammatory responses upon encounter to helminths. And to ask these questions, we actually used a different model of helminths. In this case, I used infection with Schistosoma mannsoni, which is a waterborne trematode that quickly migrates towards the tissue following different cues such as temperature or chemicals in the skin. This parasite actually causes chronic infections in about 250 million people worldwide. And in many cases, these infections are highly debilitating with high reinfection rates. Therefore, understanding this biology can also offer solutions for a big public health concern. Now, what's actually interesting about this infection is that after people get infected with this parasite, they don't really develop a strong inflammatory response, as you can see here on this picture in the center. And this is especially evident when you compare with the rash that is caused by other types of parasites that is actually known as swimmer's itch, in which in this case, the parasites cause this intense inflammation and are quickly eradicated in the skin. However, in the case of Schistosoma mannsoni, there is not much inflammation or itching, and this is actually associated with the fact that this parasite is very efficient to quickly migrate from the skin into the blood circulation and establish the systemic and chronic infections that I just mentioned before. So with this idea in mind, we actually hypothesized that perhaps schistosoma has evolved to suppress the induction of these itch responses to go unaverted and don't activate these systems. And we actually shown in my recently published studies that schisto indeed cannot only block itch, but also a specific subset of neurons that induce this type of behavior, the ones that express MRGPRA3. We can actually track the activation of these cells in the lab in an in vitro system in which we can identify these cells by the expression of the tomato reporters. And I'm showing you here quickly a calcium assay in which each line represents the activation of these cells. You can see that when we expose these cells to just vehicle or PBS, we don't really see much calcium influx, but quickly when we expose them to the Schistosoma mannsoni extract, these neurons can rapidly respond to these signals within seconds, showing for the first time that indeed our neurons can directly respond to these signals in vitro. However, when we expose them to the canonical ligand, for example, here on the left, these neurons that have not seen any schistosoma compounds, they can quickly respond to their canonical ligand, showing this robust calcium influx. However, this initial response induced by schistosoma seemed to prevent their activation to their canonical ligand. Again, reinforcing the idea that indeed schistosoma has evolved to suppress not only the behavior, but also the activation of these itch-inducing neurons. So then we ask why. Why would a parasite want to suppress the activation of itch neurons? And then we use different models in the lab, for example, optogenetics that are done by exposing blue light or blue laser in different areas of the body, in this case in the ear, to activate these neurons in genetically modified mouse systems. And we found that activation of these A3 neurons actually results in the secretion of soluble mediators such as neuropeptides and other neurotransmitters too, that are rapidly sensed by macrophages and dendritic cells in the skin, specifically by receptors expressing their surface such as RAM1. And the signaling of these molecules into macrophages opens the specific areas in the chromatin, allowing the expression of pro-inflammatory cytokines such as TNF and IL-1 beta. These signals are then sensed by the epithelium, and they are important to store these protective responses against the parasite that can either prevent the entry of the parasite into the skin or also control the dissemination of the parasite from the skin into the lungs. So this was the first time that we were aware of that we actually showed that a parasite can suppress the activation of itch-inducing neurons, because these same systems are actually really important to start host protective responses against this parasite. And in a follow-up study, we also showed that schistosoma not only blocks itch, but it also blocks pain, and specifically the thermal pain induced by other types of TRPV1 positive neurons. And again, in this case, it seems to be because TRPV1 neurons are able to mount IL-17 mediated responses that recruit neutrophils and monocytes into the skin that are able to fight this infection and prevent the dissemination again from the skin into the lung. suggesting that Shista has evolved multiple ways to target the activation of these sensory neurons in the skin. And with this in mind, I actually want to propose this concept of sensory immunity, in which we propose that neurons actively participate in the early recognition of pathogens that try to enter the mammalian host, for example, from the skin. This direct recognition of pathogens can quickly be sensed by sensory neurons, and they send these signals to the central nervous system, where they're interpret as these annoying reactions such as pain or itch, but they are also really important to activate those skin resident or tissue resident immune cells to be able to promote immunity against these pathogens. And future directions of my lab will actually address how these interactions between different species of parasites and neurons occur, and how they alter their functions, as well as the behavior that they evoke, as well as how we can actually exploit these mechanisms to promote efficient inflammatory responses that are able to contain this parasite, or even control the inflammation in other settings of excessive inflammation. And with that, I want to thank Dabrowski Herbert, my first postdoctoral mentor, especially Camila Napoli, my master's student. That was essential to get many of these experiments done, as well as my second postdoctoral mentor, Wenqin Luo, in the neuroscience department at Penn. The collaboration in different institutions, the funding from private as well as the federal funding, as well as these opportunities for career development, such as the Next Generation Faculty Symposium. Please feel free to scan these QR codes for a video abstract of these experiments, or to send me any questions you have through email. Thank you.